Welcome everyone. It's a great pleasure to give this online presentation, and I would like to thank the organizers for their kind invitation. I am Sandra van Aert from the EMAT Electron Microscopy Group of the University of Antwerp, and the aim of my talk is to show strategies for quantifying 3D atomic structures of nanomaterials and their dynamics using scanning transmission electron microscopy. In this talk, I will first briefly explain procedures for counting the number of atoms along the viewing direction. Then I will show a method that can be used to balance atom counting reliability versus the risk for structural damage by optimizing the electron dose. And I will show how one can quantitatively detect atoms from low-dose images. Next, we will see how those atom counts can be used to get the 3D atomic structure. And finally, I will come to recent progress, enabling us to track 3D structural changes over time using so-called hidden Markov models. Now, we make use of scanning transmission electron microscopy, and in a quantitative framework, the aim is to extract precise numbers for the unknown structure parameters, including the atom positions, atom types, and number of atoms from the experimental images. And therefore, we make use of statistical parameter estimation theory. Now, the key concept of this theory is the use of a parametric model to describe the underlying observations. And since ADF STEM images are peaked at the atomic column positions, each intensity peak is modeled as a Gaussian function of which we need to estimate the height, the width, and the position. Now, the estimated parameters correspond to the model best matching the underlying observations. The estimated position results in quantitative measurements for the atomic column positions, and based on the height and width, the volume under each Gaussian function can be estimated. This is a so-called scattering cross-section, which for STEM imaging is very sensitive for the atom type and number of atoms in a column. Now, in mathematical terms, this procedure can be summarized as follows. The experimental image pixel values are described by a sum of Gaussians, and the unknown parameters of this model are estimated by minimizing the least square sum. This optimization involves an iterative refinement in a huge parameter space. And it is important to notice that this algorithm takes overlap between neighboring columns into account. And this is particularly important for closely separated atomic columns. Much work has been done by team members over the years to implement this parameter estimation algorithm into our open source software, which is called StatStem. The software can be downloaded from the following link, together with a user manual. And more details can be found in this ultramicroscopy paper. Now, the estimated scattering cross-sections can now be used for atom counting, and the most straightforward manner is to directly compare them with simulated scattering cross-sections as a function of thickness. Here you see a library of simulated scattering cross-sections as a function of the number of platinum atoms that can be used to assign the number of atoms for each atomic column. Now, alternatively, we developed a statistics-based method, and here you see the same platinum particle as on the previous slide, and the question is again if we can determine the number of atoms in each column. Now, in the statistical analysis, we first estimate these scattering cross-sections, and these scattering cross-sections increase with increasing number of atoms. So ideally, the histogram of scattering cross-sections for all atomic columns in this image would consist of isolated components, where a step in intensity in the horizontal direction corresponds to an extra atom in a column. Now, if the scattering cross-sections for the orange-colored columns correspond to the 7th component, we would then assign 7 atoms to these columns. In practice, however, the components are smeared out owing to a combination of experimental detection noise and the environment. And the question is then how to determine the number of components and their locations. And therefore, we use a so-called Gaussian mixture model analysis in combination with the use of an order selection criterion. Here we find the presence of 10 components, suggesting that the number of platinum atoms varies from 1 up to a maximum of 10 atoms. Now, all columns whose scattering cross-sections correspond to the first component will be assigned 1 atom, if they correspond to the second component, 2 atoms are assigned, and so on. And in this manner, we obtain a map reflecting the number of atoms for each atomic column. Now, the accuracy of the method can be tested by comparison with simulations, and the precision is set by the overlap between neighboring components. In this example, we conclude that we have a probability of 20% to miscount the number of atoms with 1 atom, and a probability of 80% to correctly assign the number of atoms. Now, an important challenge is to balance atom counting reliability and structural damage by optimizing the electron dose. And therefore, we have analyzed a series of images of a platinum wedge as a function of electron dose. And here you see an image for an electron dose of only 1050 incident electrons per squared angstrom, from which we estimated the scattering cross-sections. However, the presence of electron shot noise causes severe overlap in the distributions of scattering cross-sections related to columns of different thickness. And this results in an atom counting error of plus or minus 2 atoms. This is also clear in this 3D atomic model, where we have placed the atoms symmetrically around a common plane. The limited atom counting reliability gives the impression of a rough surface. If we increase the electron dose, then you can see that the overlap between neighboring components is reduced, and that improves the atom counting precision to plus or minus 1 atom. Also, the 3D model now shows a cleaner surface. When further increasing the electron dose, one can imagine that the atom counting reliability improves, but on the other hand, the probability for structural damage will also increase. And the question is then how to optimize the electron dose. And therefore, we developed an analysis of variance method. And if we want to investigate if there are structural changes as a function of time, then it is straightforward to look at changes in the scattering cross-sections between 2 consecutive image frames. Without going into the mathematical details, an analytical expression can be obtained, and it can be shown that Poisson noise, scan noise, and structural damage contribute to the variance of the difference between scattering cross-sections. And those 3 terms are inversely proportional, constant, and linear with electron dose. You can also see that this last term depends on the cross-section for atom displacement, which can be modeled following the work of Ray Egerton. And this term depends, among other parameters, on the atomic number Z and the activation energy for surface diffusion. This model can now be validated experimentally, and here you see experimentally measured variances of differences in scattering cross-sections between 2 frames as a function of incident electron dose. We can now estimate the unknown parameters of our analytical expression and make a comparison with the theoretical values. And we observed that the estimated activation energy for surface displacement of 1.09 electron volt is in close agreement with the theoretically derived value for surface displacement on a 110 platinum substrate. Now that we have validated our theoretical model, it can be used to determine the optimal dose by minimizing the variance as a function of dose, and that results into this simple expression. Now, this closed analytical expression can be used to predict the optimal electron dose before conducting the experiment. Now, if we consider surface displacement of platinum atoms on a 100 substrate, then you can see that the surface diffusion energy decreases, resulting into lower values for the optimal electron dose, whereas at the same time, the probability for beam-induced surface diffusion increases. Both the atom counting precision and the risk for damage can be reduced when reducing the incident electron energy. And moreover, for thin samples such as graphene, a reduction of the inner detector angle may significantly help to detect the presence of, for example, nitrogen on graphene and to avoid surface diffusion. If our ultimate aim would be to observe hydrogen atoms on graphene, it will become hard to detect hydrogen in the presence of scan noise. even under optimal conditions for the electron dose and acceleration voltage. However, if future developments help us to overcome scan noise, then this theory predicts that we should, in principle, be able to observe hydrogen, although the contrast will still be very weak. And that brings me to a method that we introduced to identify single atoms in an objective manner. Here, for example, you see an experimental image of graphene. And in the absence of a priori knowledge, it is very subjective to determine the number of Gaussian peaks that should be refined. One can fit models with increasing number of carbon atoms, and you can see that the fit with the underlying data improve if we add more Gaussian functions. And some people will like maybe this model more than, for example, the model that appears now, but this is all very subjective. And if we keep adding peaks, then at some point there will be overfitting. And to overcome this problem, we introduced the so-called maximum a posteriori probability rule, or MAP in short, which can predict the probability for a certain number of atomic columns given the experimental data. And more details about the derivation of this MAP rule can be found in those papers. Here, for example, you see the evaluation of the MAP probability rule, predicting in this case that the presence of 63 atomic columns is most likely. And the corresponding refined model clearly shows the expected graphene lattice, which has here been obtained without using prior knowledge concerning the expected atomic structure. Now, from the atom counts, the goal is next to get an idea of the 3D atomic structure. And back in 2011, we were able to obtain an atomically resolved 3D reconstruction of a silver particle embedded in an aluminum matrix. And to achieve these results, we acquired high-resolution ADF STEM images from which we counted the number of silver atoms in each column. And this procedure has been repeated from an extra viewing direction, and by combining atom counting results of the same particle from those two viewing directions, we could then determine the 3D atomic structure using discrete tomography. Now, when studying ultra-small germanium clusters, alternative methods are needed. And from this movie, it is clear that during electron irradiation, the cluster rotates and may show structural changes. Discrete tomography cannot be used in this case. And therefore, we built an initial 3D model based on atom counting, which was then fully relaxed using ab initio calculations. When investigating larger structures, such as this superstructure, which is formed by lead selenide quantum dots, a similar methodology can be used, where instead of ab initio calculations, a Monte Carlo approach for minimizing the total energy of the structure is used to determine this 3D atomic structure. And this methodology of retrieving the 3D atomic structure from a single image opens up new prospects for analyzing in situ experiments. However, applying a similar methodology for images acquired in a gas environment is far from straightforward, because the signal-to-noise ratio and contrast gets significantly worse when imaging particles in a gas cell and in the flow of a selected gas. Here you see images of a platinum nanoparticle on a 25 nanometer thick silicon nitrogen bottom support compared to an image of a similar nanoparticle in a gas cell in vacuum, as well as in a gas environment. Moreover, those images have been acquired at high speed with a scan time of only 1 second for each frame, resulting into effects of kilohertz megahertz frequency distortions, which are very strong and often highly correlated along the fast scan direction. To overcome this problem, we trained a neural network to correct for fast scan distortions, jitter distortions, Poisson noise, and thermal noise. And therefore, we used simulated images, including a wide variety of crystal structures, elements, orientations, microscope parameters, amorphous layers, and so on. In particular, 10 million training images and 500,000 validation images have been used. Here you see the effect of the neural network when applied to an experimental image, which can then be used as an input to estimate scattering cross-sections using parameter estimation and to reliably count the number of atoms. Here you see the 3D reconstruction in a vacuum, but more interestingly, we can now follow changes of such particles under the flow of hydrogen and oxygen gases in this example. We switch the gas flow once more, and this enables us to quantify refaceting of those platinum nanoparticles with atomic resolution during various cycles. In this example, we obtained the 3D atomic structure by analyzing the underlying images frame by frame. And one of the challenges now is to investigate if we can enhance the reliability in the results by modeling also the dynamics between successive frames using a so-called hidden Markov model. Now, this hidden Markov model depends on a set of parameters, including initial probabilities, which define the probabilities for a column to have a specific number of atoms in a column. In the simplified example, the probability to have one atom is twice as large as the probability to have 2 or 3 atoms in a column. Now, the number of atoms in a column are the so-called hidden states, which we cannot measure directly. We observe them indirectly through the scattering cross-sections obtained from the ADF STEM images. Now, the emission probability defines the probability of these scattering cross-sections. Given a certain number of atoms in a column, this is described by a normal distribution function. And from this, we can see that for one time step, the hidden Markov model is in fact equal to the Gaussian mixture model introduced in the statistics-based atom counting procedure. Now, instead of analyzing a time series of images frame by frame, we will also estimate transition probabilities, which in a sense model the dynamics of the nanoparticle. It defines probabilities for a given column of atoms to, for example, lose or gain an atom. And the assumptions of the hidden Markov model are that the transition probabilities do not change over time, and that the next state only depends on the previous state, but not on the states before that. Now, using a maximum likelihood estimation algorithm, the goal is then to estimate the initial, emission, and transition probabilities. Now, the transition probabilities can be represented in the form of a matrix, with on the x-axis the number of atoms in a column at a specific time, and on the y-axis the number of atoms in the previous time frame. Now, a unitary matrix therefore implies no structural changes, whereas the presence of off-diagonal elements indicates that the structure changes over time. Once the parameters of the model are estimated, the so-called Viterbi algorithm is used to indicate the most likely state sequence. Using simulations of a hypothetical platinum nanoparticle, which is changing over time, we investigated the performance of the hidden Markov model as a function of electron dose. The percentage of correctly assigned atom counts is represented when using the hidden Markov model or when using a frame-by-frame analysis. And this clearly shows that the hidden Markov model outperforms a frame-by-frame analysis. We analyzed a time series of experimental images of a platinum wedge recorded under conditions where no severe effects of beam damage are to be expected. And here you see the number of atoms estimated using the hidden Markov model and using a frame-by-frame analysis. When comparing the transition probabilities obtained using both methods, it is clear that the transition matrix estimated from the hidden Markov model indicates smaller and more smooth structural changes over time as expected, whereas a frame-by-frame analysis interprets intensity changes related to limits in the attainable precision as structural changes. We have also analyzed the time series of images of a platinum catalyst nanoparticle using the hidden Markov model. And here you see changes in the number of atoms over time. From these atom counts, we see that while the total number of atoms remains constant, the nanoparticle loses its faceted structure under the electron beam. And from the transition probabilities, we estimate a probability of 4.6% for a surface atom to move to another column. And when taking into account the electron dose, an experimental value for the average cross-section for surface diffusion could be measured. And with this last example, I come to the conclusions of my talk, in which I have shown how quantitative STEM can be used to count a number of atoms with single atom sensitivity. We can now also predict the optimal dose by balancing atom counting reliability and the risk for damage, and detect single atoms from low dose images. From the atom counts, we can get the 3D atomic structure and track the 3D dynamics using hidden Markov models. Finally, I would like to thank everyone involved in the different research topics shown throughout this online presentation. Of course, I would like to thank the funding agencies, and I would like to thank you for listening.